All right, so my name is Ian Leifer, and I'm going to be talking about my thesis, which is called Symmetry-inspired analysis of biological networks. My advisor is Professor Hernán Makse at City College of New York in New York. And I'm going to be just giving a talk that's going to be about 1 hour, just covering all the research that we've done, and I'm going to give all the references at slides. So if you have any further questions, you can look into the references, or you can email me directly. You can email me at ianleifer93@gmail.com, or you can find me on my website. So I'm gonna first give an outline for the talk. So first, I'm going to give an introduction. I'm going to talk about what are the symmetries that we're going to be, looking at. I'm going to talk about why symmetries are important, and I'm going to talk about some applications in biology. And after that, I'm going to introduce vibration symmetry. And using this vibration symmetry, I'm going to introduce fiber building blocks and talk about their computational role. After that, I'm going to talk about how vibration symmetry leads to synchronization in genetic networks, and show some experimental results there. And I'm going to wrap it up. Excuse me. I'm going to wrap this up with the symmetry-driven network reconstruction method that we developed, and I'll describe it. And I'll finish with conclusions and outlook. So, introduction. So what are the symmetries that we're going to be talking about? Well, the symmetries that we're going to be talking about are the symmetries as they're used in their colloquial sense. So symmetries are such transformations of objects, of an object that leave an object unchanged. So it's widely used in art, it's widely used in architecture. So for example, in Delhi, there is this lotus temple here, and it's, in this picture, it's symmetric along this middle line. So if we consider the left side and the right side, of this middle line, they're absolutely symmetric. So if we reflect the object along the middle line, it's going to stay unchanged. And more geometric examples are, for example, here a square. So a square can be rotated 90 degrees, or 180 degrees, or 270 degrees for that matter, without while being unchanged. Now another example here is a rectangle, which can be reflected along x or y axes. So symmetries actually have really wide applications in physics. They lay in the foundation of the Standard Model. They lay in the foundation of Lagrangian mechanics, of Newton laws, in some nuclear and atomic spectra, and they just have wide applications all across the physics. But despite this success, they didn't find wide applications in biology. So we ask why. Well, commonly in biology, it's common to think of symmetries as automorphisms. So in biological networks in particular. So in biological networks, automorphisms are such transformations of graphs that leave the graph unchanged. So we can think again, going back to the reflection symmetries, we can think about reflection along this middle line that takes all the left side into the right side and the right side and the left. So it just reflects along the middle line and leaves the graph unchanged. So, it's easy to see that this graph is symmetric, or along, this horizontal line or any of these diagonal lines. So these are all automorphisms of this graph. So the problem with these automorphisms is that they're actually unstable. What it means is that small changes in the network can just break these automorphisms, can just drastically change the automorphism group. So if we remove this one edge here, none of the automorphisms from before are still automorphisms. So these graphs stop being symmetric. So just one change in one link can break that. And all biological networks are based on real data, so this is not perfect. And moreover, the link don't even have to disappear entirely. It can only just change the weight a little bit, and the same effect is going to happen. So this is problematic. So one of the ways how we propose to deal with this is by considering fibration symmetry. And fibration symmetry is much less prohibitive or much less strict than automorphisms. So first, I'm going to introduce input trees, and then using input trees, I'm going to introduce the fibration symmetry. So input trees. Let's consider this circuit here that's coming from the gene regulatory network of E.coli. Each node here is a gene, and interactions between nodes are transcriptional regulation. And there are two types that we consider here. One is activation, and another one is repression. So activation is shown with black arrows, and repression is shown with red arrows. So now we can look at the k in of each node. So we can consider this red node here. So it receives from itself and receives from green node. So this is k in. This is also called the input set of a node. Sometimes, just incoming edges of a node. So we take this red node, it receives from red, from green, so from itself and from the green node. We can do the same here with this other red node, it's also red and green. And we can do the same thing with green nodes here. So far we defined k in. So now input trees or k in or input sets. So to define input trees are input sets of input sets of input sets taking infinite amount of time. So in particular, we take this red node here, we move it here, so we get input sets, and we take input set of input sets, set of input sets, and so on. So we do the same thing here with the green node. And then we do the same thing here with this red node here. So we take red and green, etc. And we can build input trees. And we see that, we can build this, red input trees. We can also do this with green nodes. And before I move on, I also want to define the branching ratio on the input tree. So in order to define the branching ratio, we need to define the index that counts the number of nodes on every layer. So we call this AI. And this is just the number of nodes. And the first layer is 1, and 2 layers 2, 3, 4. So this AI counts the number of nodes on every layer. And then we can find branching ratio is AI plus 1 over AI for I going to infinity. And in these two cases, it's obviously equal to 1. This is a branching ratio that we're going to use a little later. Now, why is this important? Well, if we look at this small subnetwork here, we see that there are no obvious automorphisms. So we can't just reflect along this middle line because of the self loop. We can't reflect, obviously, along this line or any variations along this line. We can't reflect because of the directionality of these edges. So there are no automorphisms. But if we look at the input trees of two red nodes, we see that they're isomorphic, or they have the same topology. So this is the it demonstrates the isomorphism between these two trees. And this is actually very important because what it means is that these two nodes receive the same information from the network. So it's been shown that that nodes that receive the same information from the network have a certain property that I'm going to talk about later. And these nodes are called fibers. So we can break this graph into fibers. So there is a red fiber, consists of two nodes. There is a green fiber that consists of all these nodes here, and there is a blue fiber. So you can see that input trees are different. Here, these two are different because of this repression link. So we can break this graph into fibers. So these fibers are there, and they actually have very interesting connection with fibrations. So what are fibrations? Well, fibrations are maps between two graphs. So fibration P here is a map between two graphs. So this graph is G, this graph is B. So this graph is called base. And this transformation satisfies the property that's called the lifting property. And what it means is that it collapses all the nodes in the fiber. So it takes all the nodes that are in the same fiber, collapses them, and the new reduced graph has the same topology. I'll show why I'm saying it in quotes. Has the same topology as the original one. So if you look at the red node here, so red receives from red and from green, and red here also receives from red and from green. And then here, green receives only from green, and then here is the same thing. So the topology is preserved in this way. So input trees or input sets of fibers are preserved. So this is such, symmetry fibration is such transformation that collapses fibers. Now, here's just another example. When we collapse these two nodes like this, and there's an outgoing link, that's what happens to it. So why is this important? This is important because these networks, or any network, or maybe not any network, but a lot of networks, especially in biology, describe certain dynamical systems. So this happens with the set of admissible ODEs. So we can consider this network here on the left, for example, that has 5 nodes, and there's a 5 differential equations that describe their dynamics in time. So each equation, let's look at them a little closer. So there is a term that depends only on the node itself, on the dynamics of the node itself. So x1, there is a term that depends on x1. So k of x1 is just some function. It doesn't have to be minus here. I just shows it to be minus. And then there is a coupling term, and this coupling term depends on the dynamics of the nodes that send input to node 1. So node 1 receives from node 2 and from node 5. So here you can see there is a function g of node 2 and node 5. So two functions, coupling terms are the same. So, you know, just note that like coupling has to be the same for all the inputs. And we combine these coupling terms here by multiplication, but actually we don't have to multiply. It can be summation, you know, it can be some other operation as long as it can combine, you know, a few different variables in this case. So, you know, we can do the same thing here with node 3 that receives from 4 and 5. You know, you can see that, you know, here is the same thing, function of 3 and then 4 and 5. So ODEs that respect the network structure in this way are called admissible ODEs. Now, what these people in these few papers found is that there is a connection between symmetry fibrations and the dynamics of the network. And in particular, they saw that existence of these symmetry fibrations leads to synchronization. So it's actually fairly easy to see this. So if we consider the differential equations that correspond to the original system, so this bigger graph, we take these differential equations that describe it. So x's are nodes in red, y is a node in green, and z's are nodes in blue. So we can write differential equations. I took specific forms of functions k and g. Here I took k as the decay, just minus alpha x, and g is this theta function. I took it for simplicity, and you know we're going to do some simulations. So these are the functions that we use in our simulation. It can be much more general. It can be anything else, but you know here for the sake of having a specific function, that's what I did. So we can write these differential equations, and now we can write differential equations that describe this reduced network. So it's called base again. Base again. So this network has only 3 nodes, x, y, and z, and this is the dynamics. So now let's consider the solution x, y, z of the reduced network. So x of t, y of t, z of t. And if we plug in x of t, y of t, z of t in such a way that all the x's are equal to x and all the y's are equal to y and all the z's are equal to z, if we plug this in this equation, we will see that all these equations just collapse into this small set of 3 equations, which is great, which means that this solution is a solution of this system. So what we see is that existence of this symmetry fibration leads to the existence of the synchronous solution for the original network. Now, okay, I mean, this solution exists and it's great, but the question is, how often does it happen that this solution is, you know, that the network actually takes this solution, you know, and how stable it is. So like how big is the basin of attraction of this solution and how stable it is? Well, this question turns out to be pretty complicated, and there is no general answer for any, you know, that can say, oh, for any type of function, for any type of network, you know, this is the answer. There is no such answer. This is a big field of study. This reference here is one of the more recent ones that talks about it, and this is a big field of research. We, you know, here we don't give any general solutions. We just base our conclusions on some simulations that we did. So here we did some simulations in genetic networks, as I said, you know, with theta functions, and we saw that synchronization does indeed happen, and the nodes stay synchronized. And we also, you know, I'll show it in a future slides that, you know, we did it for a few different forms of equations and different variations. You know, these directed networks in directed genetic networks, and everywhere we saw synchronization. And we also saw this in the experimental data. So we took experimental expression data that we found online, and we saw synchronization. So here we see a green fiber here from this network. So these green nodes do synchronize with each other, and we see this for all the other fibers. So we see the high correlation of side fibers. And I'm going to have like a whole, you know, section on this. But yeah, so, you know, pretty much just to conclude this piece, I'll say that nodes and fibers synchronize in the networks that we considered, seem to synchronize based on our computational and experimental results. Now, before I go into results, I'll just briefly talk about the algorithm of how to find fibers. So this is also like a wide area of study in computer science and dynamical systems, and there are many different algorithms. we adopted these algorithms to fibers because the algorithms are for balanced coloring, which is almost equivalent to fibers. What we are considering is slightly different. There is a slight variation. So, but you know, I'm here going to give simple examples where it doesn't matter. So this is the whole story here is based on the input set color vector. So input set color vector is the vector that corresponds to each node. It's n dimensional, where n is the number of colors of the graph. So here, you know, in this graph with one color, the vector is going to be one dimensional. And each entry of this graph corresponds to number of nodes in the input set of a node of a given color. So if we consider the node 1, and input set is going to be node 2 and node 3, we can see that there are two nodes of the white color in the input set of 1. So we see that 1 has the input set color vector equal to 2, which is one dimensional, or you know, 5 only receives from 4, so only 1, and 8 is 0. So what we want to find is we want to find such coloring of the graph that is balanced. So balanced means that node of the same color have same input set color vectors, and node of different color have different input set color vectors. So this coloring is obviously not balanced because 4 and 5 are of the same color and they have different input set color vectors. So in order to get to the balanced coloring, we can go through the process called refinement, partition refinement. So this is an initial partition where all of them are of the same color. We create a new partition here based on the input set color vectors. We recolor the graph, find the new input set color vectors, go through this again and do it again, and then ultimately we get the final coloring that is balanced. So in which the nodes of the same color have same input set color vectors, and nodes of different color have different input set color vectors. So for further reason, you can see these two papers. These algorithms, you know, the variations of this algorithm are available on our GitHubs as R packages and as a Python package for you to use. So now using this Fibration symmetries, we can define fiber building blocks, and the variation of fiber building blocks is called symmetry breaking building blocks. I'm gonna talk a little more about that later, and I'm gonna talk about that computational role. So what is a fiber building block? Well, fiber building block, consider this graph here that has one fiber of the green color. So we can find a block that corresponds to this specific fiber. So fiber building block is an induced subgraph that consists of all the nodes in the fiber, so all the green nodes here, all the regulators that send input to the fiber, so this one regulator here, and all the nodes that belong to the shortest loop, including a node in the fiber, if a node in the fiber is a part of the loop. So in this case, this green node is a part of this loop here, and we include this node here in the building block. So we can find fiber building block here, this is it, corresponding to this green fiber. And we know that like there is a longer loop here and it's not included. So the idea behind this is that if we choose fiber building blocks in such manner, we can study the dynamics of this building block separately from the dynamics of the rest of the network. So dynamics of this fiber is only influenced by this dynamics of this closest part of it. And then the rest, we can study the rest of the network separately and then come back here. This is going to be clear later why this is important. So, you know, another example here is this circuit from before. So if we consider this circuit, it breaks into 3 building blocks corresponding to 3 colors. By applying this analysis, we can find all kinds of different topologies of building blocks. But it turned out that actually we can classify all these different topologies using only 2 numbers, which we call fiber numbers. So the first fiber number, and these fiber numbers characterize the input tree of a fiber in this building block. So if we consider, for example, this green fiber here, it only consists of all the green nodes because no other nodes send inputs here. So input tree is just going to be this infinite chain, just one node on every layer. So in branching ratio as before is equal to 1, as we talked about before is equal to 1, and there are no external regulators. So 2 numbers, n is branching ratio and L is the number of external regulators. So this circuit is classified as 1, 0. And similar analysis can be done for the FFF building block where there is one external regulator, there is a self loop, and input tree looks like this. So branching ratio is equal to 1. and there is one external regulator. So this is 1, 1. And here, when we have finite input tree, we agreed that branching ratio is equal to 0. So this building block is classified as 0, 1. So now using this analysis, we can go into, or not we can go, but we actually did go in this paper. We went into like 400 or 500 different building blocks. And in these building blocks, we, sorry, the different networks. And in these networks, we identified building blocks and we classified them. So we saw that in all of these networks, building blocks are, I think we saw like 1 or 2 building blocks with n equals to 3, but almost all building blocks had n equals to 0, 1, or 2. So, you know, that's an interesting result here. So, you know, and different values of L. So I give some examples from E.coli on this slide. So, you know, 0, 1 as before, 0, 2, you know, 1, 0, 1, 1, you know, etc. So there are a couple of building blocks that are important, and I want to kind of stop, you know, just focus on them for a second. So this building block is called bifan. So this building block is an automorphism. Easy to see that it's symmetric along this middle line, and this building block is 0, 2. So this is a famous building block that was introduced by Alon and his collaborators in this 2002 paper, and it had like a major impact on systems biology, and our method also allowed to identify this same building block. Now, another building block that is very similar to the one introduced by Alon is this one. We call it FFF, feed-forward fiber. So it's symmetric. It's not an automorphism. You know, we can see that there is no way to reflect anything here without changing the structure. So it's not an automorphism, but it has fibration symmetry. And it is similar to the building block introduced by Alon. It's called FFL, and FFL doesn't have this one loop here. So FFL looks like just these 3 nodes without this self loop. So here, and you know, it's easy to see that FFL is not symmetric and FFF is symmetric, because without this one self loop, there would be no symmetry in this circuit. So that's the circuit that we identified. Another circuit that we identified that is important to mention. Now, so far we talked about the building blocks with integer branching ratios, but what we found is that in building blocks that have self loops, or not the self loops rather, but the loops with the regulators, we see that there is branching ratio is not integer anymore. So, and this is pretty interesting. So, for example, this circuit here, this circuit from E.coli here, it starts, the input tree starts looking like a 1, 2 building block, and then one of the regulators receives back from the fiber, as you know, as was mentioned, and then there is this whole input tree here, you know, like the same input tree is attached here recursively. So, if we look at the number of nodes in every layer, we will see that number of nodes in every layer follows this sequence, which is a very well-known sequence, and it's the Fibonacci sequence. So branching ratio in this case is going to be equal to golden ratio. So this is a special type of building block for which branching ratio is not integer. And we found a few examples of this in which there are longer loops here, as can be seen. And these longer loops create, you know, these different variations of trees. And in general, for the loop of length D, we can see that this is going to be the input tree, and the sequence is going to look like AI is equal to AI minus 1 plus AI minus D, which is called generalized Fibonacci sequence. And the branching ratio is always going to be non-integer, and there is a solution for this branching ratio for, you know, for different Ds depending on the D. So these are fiber building blocks, and these building blocks represent a certain short-term memory in a way. Like it's a memory, like, so the state of this node is kind of preserved here, here, and here, and then it kind of comes back. So it takes a little bit to go through this loop, but there is a memory that in this circuit, like a short-term memory. So another type of the circuit that we want to consider is the multi-layer circuit. So multi-layer circuit is this. So if we consider this small part of it, so this small part looks like a 0-1 building block as before. But there is another node here that is synchronized with these nodes. So and it's you know four steps away in the network, so it's pretty far. And so if we look at the input tree here, this input tree is like a combination of two input trees. The first part of it is, it looks like 0, 1. So just, has this like 0, 1 structure. And then the second part looks like 1, 1. And we can say that, we say that this building block is a composite building block, and it's called 0, 1 plus 1, 1. Now, let's talk about the dynamics a little bit of these, these circuits. So the simplest circuit that we want to consider is SAT-FFF. So this building block, all the links are activation links. X of t we just consider as like external signal in time. So we only consider the dynamics of y and z. And it can be written like this. And it's easy to see that if x is less than k, then this whole right side is 0. And then both y and z are just two decaying equations that decay to 0. And then if x is higher than k, then and y is higher than k also, then this right side is just going to be equal to 1. And they're both going to go up to a different value, which is also the same value here. And that's exactly what we see in simulations. We see synchronization. We see that they slowly synchronize together, and they stay synchronized. And they go, so we change x from above kx and below kx. So we go from 0 to 1. And we see that they stay synchronized. So they get synchronized, they stay synchronized, regardless of any external conditions. And that's, that's the expectation. So SAT-FFF plays the role of synchronization. And X is oscillating, but, in reality, it can just, if it was in one state, then it would go, Y and Z would go to one state. And if it was another state, Y and Z would go to a different state. So another circuit that is slightly more interesting is this one. So we consider just one node with negative autoregulation here. And the convenient way to think about it, just, for the visual purposes, is using the analogy to logic circuits. And it's very common in, in synthetic biology to use this analogy. So analogy is just a NOT gate. So we take the state of a node and there is a NOT gate. And then the state and time can be written as yt is equal to NOT yt minus 1, which pretty much means that that this circuit is going to synchronize. So it's going to go from 1 to 0 to 1 to 0 to 1 in Boolean dynamics. So it's just going to oscillate, and that's exactly what we see in the ODE model as well. So we see this oscillation here. So we expect that this genetic network, this small circuit with negative self-loop, will play the role of oscillation, so it can play the role of a clock. And that's exactly what we see in a bigger system. So we consider this on SAT-FFF here that has this negative self-loop part here, and then there is another edge, and then there is external regulator, and we can look at the electronic analog again that's written here with these equations, and that's what we see. So y and z start in different states. So y starts at 1, z starts at 0, and then we look at the dynamics in time. So this is time. So they start at 1, 0, then they go to 0, 0, 1, 1, 0, 0, 1, 1, 0, 0. So they synchronize and oscillate. So, and that's what we see in the ODE model as well. So we see that on SAT-FFF synchronizes and oscillates. So we believe that the circuits that have these type of negative self-loop oscillate, and they play the role of a clock. So far we found a biological clock. Now what we can find, let's see if we can find something else that's interesting here. So let's consider this building block consists of just two nodes in this negative self-loop. Let's consider the base of this building block, not instead of the block itself, so just one node with negative self-loop. So in biology, it's very common to think about replication. So this one node replicates into two, like duplicates itself, sorry, into two nodes, and negative self-loop becomes this negative loop between the two. And this is still a symmetric circuit, but now in reality, it's going to receive some external inputs. So there are inputs, and let's call inputs to y s and inputs to y prime, let's call them r. So these are, this circuit is a very famous circuit that was introduced in this paper that started synthetic biology. And this is the circuit, it's called, this is a toggle switch. So this circuit is analogous to toggle switch in electronics, and toggle switch is the basic memory unit. And I'm going to talk a little bit more about the dynamics on the next page, but just for now, this is a memory unit that is yeah, that's analogous to toggle switch. Now, another example here is FFF circuit. So we start with FFF, we go through the same, we collapse the base, we replicate it, or we go through duplication, and then we break the symmetry. And then we get this another circuit, which is SR flip-flop, which is another memory circuit in electronics. And the last one here is a Fibonacci, so it's a more complicated, circuit with self-loop here, which is, this circuit just has extra, loop back, and going through the same process, we get JK flip-flop. So we get 3 memory units of different complexity going through this process. So far, before we showed some oscillation, so the clock, the function of the clock, and now we're showing some function of the memory. And in particular, let's talk about just this one because it's the most famous one. So the electronic analog looks like this. And it's easy to see that it can't occupy a state of 0, 0, because if it was 0, 0, then in the next moment this 0 would become not 0, so 1, and this 0 would become not 0, so 1, and it just can't. It's not a stable state. And then 1 would become to 0, so it will have to jump very quickly between zeros and ones, and it doesn't seem to work this way. So the stable states of these circuits are states in which one of them is equal to 0 and another one is equal to 1. Then not 0 is equal to 1 and not 1 is equal to 0. And then the state is stable. And another stable state is 1, 0. Similarly. And in order to switch between the states in external inputs S and R should be 1 and 0 and 0 to 1. As shown here. So to go from 0, 1, 0 to 0, 1. Sorry. To go from 0, 1 to 1, 0, it needs to be, S has to be equal to 0 and R equal to 1, and and the opposite one as well. So what we see is that this is the long-term memory unit. So we have long-term memory, we have somewhat short-term memory through these loops, as I mentioned, and we have the oscillatory behavior, which is clock. So we can apply this whole machinery, and then we also have a reduction process that we can apply using symmetry fibrations. So using all these tools, we can do this, just to give a little more context. So this context is based on this paper that still is in preparation. I'm not gonna go deep into detail because it still is not final, but the idea is the following. So we consider this genetic network of E.coli, and it has about 800 nodes. And so this network can be reduced for a network by collapsing all the fibers and removing all the nodes that don't send back to the network, that just are external nodes that only receive from the network. So to find this, we can identify the core network that drives the dynamics of the entire E.coli network using fibration symmetries. So this network is only 50 nodes, which is much smaller comparing with a big one. And now we can study the dynamics of the reduced network and then go back to the dynamics of the big one. And reduced network actually turns out to contain a lot of symmetry breaking circuits. And that's what we find. We find, for example, flip-flops. We find quite a few flip-flops here in this reduced network. And flip-flops meaning the toggle switches in this reduced network. So pretty much the idea is we can take the big network of E.coli, we can reduce it, and then we can study the dynamics of the reduced network and then go back. And therefore, fibers and fiber building blocks provide this, very convenient set of tools to analyze any network. So this gives a little more context here. Now, I'm going to talk a little bit about what are consequences of symmetry fibrations. So as I mentioned before, symmetry fibrations have never been observed in any of the real networks before our work. So obviously there were no experimental confirmations. So we obtained the first experimental confirmation. So this is a genetic network of E.coli that I showed before. This is like a small part of it. And we looked at the synchronization or in the gene expression of these genes in this part of the network. So we see that genes inside fibers are highly co-expressed or highly correlated. So these nodes, for example, here corresponds to this fiber. And all of them are highly correlated, and this blue one is highly correlated. You can see the bigger one. So there is a high correlation, and this is the alternative carbon source. Oh yeah, okay. And another thing that's important is that by taking this circuit, so this circuit is just taken from the topology. So we just chose this one strongly connected component and all the fibers around it. And what we saw is that actually we can classify each fiber and give each fiber a biological function. So it's actually very interesting that we can just, without really knowing anything about the biology, we can just go from the topology and identify these parts that play different functions. And we did this functional analysis later, and that's what we saw. Now, also we can see that there is a correlation inside the whole hierarchy. So it happens for all kind of different fibers. So for 0, 1, we see that there is a high correlation. For 1, 1, for multi-layer, for all of them, we see the same effect as before. We see correlation inside fibers for all hierarchy. So correlation inside is high and outside is low. And we see something interesting here. So let's just take a look at this circuit for a second. So this is, So this is the data from Bacillus subtilis, and this is the data from E. coli. Okay, these are 2 bacteria. So Bacillus subtilis here. So if we consider this circuit, we actually see that all of it is synchronized. Like red nodes are synchronized with green, synchronized with black. Black is this regulated here. So they're all synchronized. Now, why would that happen? Actually, just before I say this, so this one node, it may be tempting to think that this one node is this one, and it's not. It's one of these. So just a side note. So they're all synchronized. Well, one of the explanations why they all can be synchronized is that there may be a negative or a self-loop, positive actually self-loop here. If there was a self-loop here, then all of these nodes would be of the same color. They would all belong to the same fiber. So actually what this shows is that vibration symmetries or fiber building blocks provide actually very interesting tools for predicting what links are missing. So we can predict that that based on the expression analysis that we see here, this one link can likely be present in this network. So it kind of helps experimentalists to just direct the research of experimentalists. It's another interesting application. So we want to assess the significance of these correlations that we just showed. And to do so, we're going to use the correlation coefficients. So we're going to correlate expression of pairs of genes for different conditions. So we have some data about how genes are expressed for in different conditions, and we're going to correlate genes with each other. So for example, if we consider this network, again from E.coli, and all examples here from E.coli, but actually this can be applied everywhere else. But anyway, so if we consider this network, this small piece, we see that red nodes are highly correlated with each other, and the same happens with green nodes. There is low cross correlation. And in order to look at the collective effects, we have to consider the average cross correlation. So we consider average of all of diagonal terms here using this equation. So that's exactly what we did. So we looked at the average correlation for different building blocks depending on the size, or for different fibers depending on the size of the fiber. We see here that red shows the average correlation of random sets of nodes of the same size, and blue shows the correlation of fibers. The fibers are clearly expressed higher, and by doing hypothesis testing, we saw that 70 out of 85 are significant. So most of the fibers are significant here. We did do some further analysis because we had to apply some filtering. So because there are some conditions that are not very good, it's common to I'll explain what not very good means in a second. It's common to apply filtering. So not very good means that these conditions are called not active. So filtering is based on inverse coefficient of variation. So inverse coefficient of variation is mean over the standard deviation. So we take one condition. So each column is a condition, each row is a gene. So we take one condition for the gene, and we check what's the average correlation in this condition and what's the standard deviation of this condition. And we take conditions for which the mean is higher than standard deviation, than few standard deviations. So what does it mean? It means that we take conditions for which expression values are somewhere above, like, let's say this is the 0 level, and then we take conditions in which expression is somewhere around a certain mean that's higher than 0. Because in conditions where expression is low, is just around 0, these are conditions that are not where fiber is not active. So we filter for these active conditions. And it's common that this data is taken from Columbus, and this is like it's just a very popular gene database, and that's the filtering that they do, and we kind of reproduced it here. And we did the significance analysis here, and again we see that blue line is above red, and we see that there are less fibers that are significant. It's only 30 out of 85, but actually most of the insignificant fibers are in small sizes. Like it just so happens that smaller sizes are insignificant, and we believe that it happens because this filtering just creates some bias towards the smaller sizes. Just it makes them overly correlated. So like these few are not significant, but the rest are pretty good. So these are the results that we got here. Now, the last thing I want to talk about is the symmetry-driven repairs of graphs. So the method that we developed to repair the graph. So far, we only talked about the advantages of using fibers versus automorphisms. So just considering a less restrictive symmetry, that's the symmetry on the topology rather than, well, on the local topology, let's say, rather than global topology. Now, if we consider this circuit here on the left, and these are the colors of this circuit, just again, by removing just one edge, all the colors are broken again. So red break into red and light blue, and then yellow breaks into yellow and dark green, and then green breaks into green and blue. So, but orange one is preserved, but still most of the coloring is broken. So there is again this instability. So how do we deal with this? Well, we propose this method that is called pseudo-balanced coloring. It optimizes the pseudo-balanced coloring. So we developed an integer program that what it does is this. It can find number of edges that needs to be added to a graph. So we give it a graph and we give it number of colors, and then it repairs the graph to this number of colors by adding minimal amount of edges. So what is the minimal amount of edges that needs to be added to this graph to repair it to the given number of colors? So there are a few steps to this process. The first step is we fix the nodes that are already symmetric. So we in this case, for example, we obtained that nodes 5 and 6 are already symmetric. So we fix them. And then after that, we find solutions for all different numbers of colors, different possible numbers of colors. So the lowest number of colors is going to be 1. So all of the nodes will be of the same color. And the highest will be 1, 2, 3, 4, 5, 6. So 7 colors is going to be the highest number of colors. So we find all the solutions with 1, 2, 3, 4, 5, 7 colors. And then we choose the best solution. So we evaluate these T graphs or these graphs that we get using the indices. These are certain characteristics of graph topology and dynamics that I'm going to talk about. We choose the best graph. So we consider two circuits. We consider the circuits from C.Elegans. C.Elegans is we consider the neural network of C.Elegans. And that's the worm that's fully mapped out. It has about 300 neurons. And we consider that network is just a well-studied network. And we considered backward locomotion circuit, which is only about 30 nodes, and the forward locomotion circuit. So these two circuits, we will see that these two circuits actually kind of represent two different topologies, like topological types, let's say, of repairs. So this first topological type is like the type in which the network can be decomposed into separate parts that can be repaired separately. So for example, if we look at these 6 nodes here, only 2 of them are symmetric in the original network, and the rest are not. So there are small repairs that can be applied. So there is one repair of this one edge that can fix this one part, another repair that can fix another part, and then this repair that can fix the whole thing. Or, Then another circuit that's interesting is, all these nodes here. So these nodes receive from both of these hubs. These 3 nodes only receive from the left one, and these 2 only receive from the right one. So the whole thing can be repaired by just adding a few edges, corresponding, that are missing. So this is another repair that can be obtained. Or this one is the one that, I showed on the first slide is very, it's the same repair. So just by adding this one edge, then the whole bottom part can be repaired. So these are a few repairs that we obtained for this backward circuit, or that our code was able to obtain for a backward circuit. And as we progress, going from the high number of colors where all the nodes are in separate colors to the low number of colors where all the nodes are in the same color, we see that these repairs are slowly obtained more and more. So each column is different number of colors and each row is a different repair. So ultimately, all of these repairs are combined together in the final repair. So this is a process how it goes here in the circuit that's decomposable. Now, next circuit that we want to consider is not decomposable. It's actually, well, it is decomposable, but the repairs are not decomposable. So the lower part is already symmetric here, and the upper part is not symmetric. This is the original circuit. So it can be like decomposed into these two parts, but the repairs are only going to happen in the upper part. Now, because this circuit is undecomposable, in this way, the repairs are going to be unstable, which means that the edges that are repaired are just going to be different for different number of colors. So that's exactly what we see. We see that for different number of colors, each column, the edges are different. So it's, here, these are repaired, and these, 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 they jump, they change. So, the edges repairs are different. So, but what we can see is that the graph becomes more symmetric, the more, so here we start with all the nodes being, so they're all white. White means that they're trivial, meaning there is only one node of that color. So these nodes are not symmetric, they're all white. And then over time they get, first these white nodes become red, then, these blue come in, and then so on and so forth, until ultimately almost all of them are colored aside of just one. So that's exactly what we see. So we see the graph becoming more and more symmetric. But this is the only effect we can see. But now, how can we judge, which one is the best one, especially like, how can we do it automatically? Well, in order to do this, we came up with the concept of indexes on graphs, or rather, we came up with a few indexes of graphs that can be studied in order to answer this question. So we considered a few. I think only two really need to be discussed here. So first one is the number of non-trivial colors. As I just mentioned, the trivial colors are colors of which there is only one node. So non-trivial colors, is the opposite of that, which is the color which has more than one node. It's easy to see that when we repair the graph with a low number of colors, let's say, for example, only one color. If you recover the graph to only one color, it's going to be only one non-trivial color. And if we recover a graph to a full number of colors, so if there are n nodes, if we recover it to n colors, then there's going to be 0 non-trivial colors. So all colors are going to be trivial. So it's easy. What it means is that number of non-trivial colors is going to be low on one end and on another end of the spectrum. So we hope that it's going to be high somewhere in the middle. And that's exactly what we saw. We saw that number of non-trivial colors has this interesting shape where there is a peak here in the middle, and then here is also the same thing. So these are backward and forward circuits. And we choose the best solution based on this. So we choose this green line corresponds to the best solution, and the red line corresponds to the best solution that was obtained manually. So we did some manual repairs, or rather, there was a paper where people did some manual repairs of this graph, and we were comparing with it. And here it coincides, and here it's slightly different, but without going deeper into this, they're actually pretty close. So despite the fact that these look different, they're actually very, very similar. So we can see the best repair here on the right. So the best repair, fixes this one edge, fixes these edges, and then these other edge. So it fixes some parts of the graph. And the same happens in the forward. Actually, forward one is very symmetric, the repaired one. And yeah. So we really think that the answer, like the good answer to which repairs are the best, should be based on the dynamics. So we're giving here the topological answer because that's what we found, but we do believe that the answer is in the dynamical answer, in the dynamical index. So one of the dynamical indexes that we considered was Fiedler value. So Fiedler value is this known index that describes the synchronizability of the graph. So the ability of the full graph to be fully synchronized. So ability of all the nodes of this graph to synchronize. And actually, what we want in this case is we want this value to be low because the nature wouldn't create this circuit that has very complex biological, few different functions, and just all of them would synchronize. Like it wouldn't make sense for all of these nodes to synchronize. So we want to stay in, or we want to find a solution in which this total synchronization is low, which is exactly what we do. So we see that it increases when the number of colors is decreasing, and our solution is here in the region of low total synchronization, and same thing happens for the forward circuit. But this index gives us an idea. So an idea for the future work, and that's the work we do in this paper that is under review currently. It doesn't fully give an answer to this problem, but it's a step in that direction. So we believe that the answer is in cluster synchronizability. So if there was an index that would describe the ability of clusters or parts of the graph to synchronize, we believe that this is going to be the answer. Why? Well, because we believe that this circuit, not we believe, but this circuit performs certain biological function. This circuit is symmetric. So it means that parts of it need to be are synchronized or can be synchronized. So we believe that nature has certain evolutionary pressures towards the circuits that are going to be that can create these synchronous dynamics in certain conditions. So we believe that stable synchronous dynamics, moreover. So we started some work on developing some index of that nature, and we think that future work needs to include this analysis. Again, we can't give a good answer, but we couldn't give the general answer. Sorry, but we took some steps in that direction. So I'm just going to give a few conclusions and give some outlook to wrap it up here. So first conclusion. So fibration symmetries helped us uncover new functional building blocks that are related to synchronization. So we developed these, as I showed, we found these new fiber building blocks, and they're related to synchronization. Now, some of the building blocks or functional building blocks can play a few roles. They play the role of synchronization and clock, and then there are some functional building blocks that play the role of the memory. So we found these, two types of building blocks: synchronization, clock, and memory. And yeah, so furthermore, fibration symmetry provides a novel way to analyze biological networks by reducing them and studying the reduced network. So that's what I demonstrated. So, using these advances, we can create this reduced network, and we study it. Yeah. Then the next thing I want to talk about is that, in genetic networks, fibration symmetry leads to coexpression, or rather can help detect gene coexpression patterns and can help detect some missing links, or give an idea for some missing links. And last thing I want to say is pseudo-balanced coloring, what I just described, provides an approach to network reconstruction that restores the symmetric structure of the network. And just for the future research, just a few thoughts. So first, this analysis, all of the analysis that we showed, aside of the very last part, was applied to genetic networks. And in particular, it was applied to just genetic network of E. coli. So this is obviously, a first step. So it can be applied much wider. It can be applied to any other genetic network. It can be, we applied it to bacillus very briefly, but, it can be applied to humans, yeast, all over, it can be applied everywhere. It has a huge potential. And it really is not limited to biological networks. It can be applied to any directed network. You just need those further analysis, but it can be applied to any directed network. And obviously better, there is a space for better studies of gene co-expression, because, in our studies, we just studied different conditions, but actually in reality, dynamics that we should be studying is the dynamics in time, so change of dynamics in time. And there are many different possibilities where experimental confirmations can be obtained, neural networks and just all kinds of different networks. So there is a lot of potential there. And then this pseudo-balanced coloring, algorithm that we showed here, for network reconstruction, it needs to be extended for higher sizes. So what we developed is not very fast. Let's say we have some ideas for how it can be, how we can speed it up, and we did outline them in our paper, but there is further work to be done, and it can be obviously again applied to all kinds of different networks. So all the methods that I described here have wide applications to all kinds of different biological and just directed and undirected as well to, networks in general. So that's the conclusions and just some future work. So for further reference, you can see these papers here, or you can email me if you have any questions. Thank you.